Hi students, welcome to Year 11 Biology and Module 2, Organisation of Living Things. This is video number 10 and we're going to have a little bit of a look at gas exchange in plants. Our idea here is for you to investigate the gas exchange structures in plants through the collection of primary and secondary data and information, for example, microscopic structures such as leaf structure in plants. So what are the important gases that plants require? Well, they don't require oxygen. They do require oxygen, actually. They require oxygen for respiration. But they are net oxygen producers. That is, they produce more oxygen than they actually consume. So the processes of photosynthesis, which require both carbon dioxide and water, are going to generate oxygen as a byproduct of that process. And whilst plants will use some of that oxygen in the processes of respiration in the mitochondria of their cells, they will also release excess oxygen to the atmosphere. This was a very important component and one that we've seen reflected in the history of life on Earth, particularly in phenomena such as the banded iron formations or red beds, when the concentration of oxygen was increasing as a result of these early photosynthesizers and was turning a lot of the free iron into iron oxide. The gas exchanges that occur in plants are either directly across the plant surface or via special structures in the leaf or the stem. Water moves from inside the leaf to the outside via diffusion, and likewise carbon dioxide moves into the plant from the outside to in via diffusion. So we know that diffusion is about setting up concentration gradients. As long as there is a higher concentration in one place, a lower concentration in another place, and there's the ability for those substances to move, either a semi-permeable membrane or, in the case of the stomata of leaves, just some pores, direct openings through which the gases can diffuse, then they will in fact diffuse either in the case of carbon dioxide, or out in the case of water and oxygen from the plant. The specific structures that are involved in gas exchange in plants are stomata. This is an electron micrograph that I was given some time ago for presenting actually at a science conference, Canasta 67. And it shows you a number of different stomates or stomata, 4 stomata, 1, 2, 3, 4. There's also a little pollen grain in there as well. This is from a grass bud. So you can see these are the microscopic structures, the stomata, and they are the ones that are responsible for gas exchange. The way they do that is that each of those little pores, and you can see on the electron micrograph that they're all closed. They're pretty much all closed, not much of the opening here. And that happens when the guard cells, the ones around the stoma, are flaccid. They've collapsed basically in on themselves. As these guard cells fill with water, their quite unique shape, basically, and also some reinforcing in the cell wall, slightly changes the shape of the cells as they fill with water. And what that allows them to do is to open that stomatal opening, that pore in between the guard cells. And what that actually allows is material to exchange into and out of the plant via these little pores. So guard cells, I guess, specifically controllers of that gas exchange in plants. Now, these sorts of fellas can be seen when you look through a light microscope. Sometimes you can actually see these particular cells scattered through generally the lower epidermis. So that is the underside of the leaf. A lot of the time, the upper surface would lose too much moisture if it had a high concentration of stomata. So we find that in areas where there's a lot of moisture, such as the plants that live in rainforests. But for a lot of other plants where water is a little more scarce, there tends to be a cuticle layer protecting the upper epidermis, and most of the stomata are located on the lower surface, sometimes even sunken in pits to reduce water losses. So we've already looked at the fact that the opening and the closing of the stomata are regulated by water movement, and specifically water movement into and out of the guard cells. We can make some generalizations about when stomata are primarily open or closed, And I guess the first general way to link them is that they tend to be open in the light conditions and closed in the dark. One of the main reasons for this is that the first part of photosynthesis is the light dependent stage, and in the light dependent stage we are decomposing water into protons or hydrogen ions and electrons, and they're going to be a very important part of the photosystems that take place in that first stage of photosynthesis. So that means we need lots of water, we need water moving through the plant, and as we'll look at later in this course, we'll see some of the phenomena associated with the transpiration stream that's actually pulling that water through the plant from the roots up through the xylem and out through the stoma. So in the day we get a lot of oxygen coming out and water coming out and carbon dioxide coming in. So this is important, as we said, for photosynthesis. At night, potassium tends to move out of the guard cells, and that initiates an osmotic gradient, and as a result of that, the water also will then move out of the guard cells. That'll collapse the guard cells, they'll become flaccid, and they will close the stomata. So again, these are generalizations. There are obviously going to be exceptions, and of course, particularly in an environment where if it's very, very dry, it hasn't rained for a while, water availability is low, the stoma are still not going to be completely open through the daytime either. Otherwise, the plant would just dry out. At sunrise, the photosynthesis process starts to increase glucose levels in the guard cells. And what that does is that it creates a reverse osmotic gradient to the one that was created during the night. So the movement of solute ions out had an effect to dilute basically the solution, which encouraged osmotic movement out of the cells. Now an increase in glucose is basically increasing the solute concentration. It's now going to create an osmotic gradient that's going to bring water in. The fact that water is now starting to come in is going to make these guard cells turgid. And the fact that they have thicker walls on the pore facing side, so that those little outlines where we looked at the pore, where we looked at the stoma in the center, is going to create that slightly changed structure and cause the outer cell walls to stretch a little bit more. And that turgidity is what opens the stomata and allows for exchange of gases through that structure. As I mentioned, if there is high water losses during the day, that will stimulate closing of the stomata, because the whole idea with this opening and closing of stomata to regulate gas exchange that's occurring in the plant is all about getting water up to the leaves for photosynthesis, but trying not to lose too much water out through the leaves. That can't be replaced. Gas exchange is quite a complex process. In terms of plants, this is the key one. We know that the structure of a leaf contains a large number of mesophyll cells as well, so that's cells that are in the center of the leaf that are either of the columnar palisade form or of the spongy type. And the spongy mesophyll cells still high in chloroplasts, but also are surrounded by air spaces that allow the diffusion of these very important gases to not just come into or out of the plant, but to move in contact with the cells as well. As I said, this is the microscopic level. We will have a look at the macroscopic level for plants and also gas exchange in animals coming up in future videos. Thanks for watching.